Hello, welcome everybody. It is my pleasure today to introduce Professor Mario Marconi. Professor Marconi got his degrees, licenciado and doctorate degrees from the University of Buenos Aires in Argentina. He then came here to Colorado, at the Colorado State University to work as a visiting scientist. He went back to Argentina as a professor there. He found some good graduate students and me.
Yeah.
Then he got tired of me and came back to Colorado.
And he followed.
And where he's now a professor at the engineering electrical and computer engineering department, and he's a participant of the NSF ERC for Extreme Ultraviolet Science and Technology, which is a center that also has a group here in CU in physics and in Berkeley as part of the center. And his interests include the development of tabletop extreme UV lasers and applications for those lasers, nano-patterning, interferometry, and high-resolution imaging. So, thank you.
Thank you. Thank you very much. Okay, well, it's a pleasure to be here. So, I would like to make this talk very informal. So please stop me any moment you want to have if you have a question. Okay. I use this biograph for the first biograph in my talks when I go outside for Collins. Here is almost irrelevant. I am not going to impress anybody here with the maroon belts, but I like them very much, so I leave this figure. Well, my work is, I am part of the Engineering Research Center for Extreme Ultraviolet Science and Technology. This is a center funded by NSF and has three institutions. CSU is the leading institution. The other two institutions are CU, the group in Gila with Henry Kapkin and Margaret Murnane. And the third group is in Berkeley, in the Lawrence Berkeley National Lab. These three groups coexist, and we have complementary tools to develop extreme ultraviolet science and technology, and my talk will be focused in this particular part or this particular aspect of imaging objects with very short wavelengths. So the outline of my talk first will be focused on the description of the sources that we develop in CSU. These are tabletop or what we call desktop smaller lasers. The laser is this small unit here, this small box, or this tank here. These are very compact as compared with other UV sources that are large like synchrotrons. So the first part of my talk will be devoted to explain very briefly how these lasers work. And I will describe applications with this laser. We demonstrated applications like microscopy, lensless imaging, this is diffractive imaging, nanoablation, nanopatterning, holography, plasma interferometry, and few other applications that I will not mention here. In particular today, I will focus in these three applications: nanopatterning, holography, and microscopy. So going first to describe what is the ERC, this Engineering Research Center. We focus in applications of unique new lasers. And everybody knows the importance of lasers in technology. Nowadays, lasers are very compact, very efficient, and cheap, and they have application in absolutely many fields like holography, optical storage, ranging, surgery, you name it. Lasers are everywhere. And these are very well developed applications for visible and infrared lasers. However, there is a region in the spectrum, the region between, I would say, electron energies, photon energies, about 10 to 500 electron volts, that is almost unexploited. This is called the UV extreme ultraviolet or soft X-ray region. This is unexploited, and there are few reasons why this is a region unexploited. It's because sources in these regions are very hard to fabricate. However, this region is very useful. Why? Well, because the wavelengths of these photons are between, I'd say, 10 nanometers to 100 nanometers. So they match exactly to nanostructures. So they are, I would say, perfectly suited to work with nanostructures and nanotechnology. And people realize that, and these are examples of applications that ourselves and our colleagues from Berkeley has demonstrated. Like, for example, these two are demonstrated with a synchrotron light. These are imaging of printed circuits. These are a tomography of living cells. These are applications that we demonstrated in Fort Collins with a nanoscale imaging or a nano machining or nanoablation, interferometry, or nanopatterning. The idea and the usefulness of these photons in this region of the spectrum is recognized everywhere. So people build these big factories, these big photon factories. These are huge machines. This is a synchrotron in Berkeley. This is a recently commissioned free electron laser in Hamburg. To have an idea, this is a building, these are cars, right? So this is a huge facility. In the case of the synchrotron, it can accommodate like 25 beams. This one is a single beam facility. It's a laser that is tunable and goes to 15 nanometers or a little lower, but only one experiment at a time. And this here is a linear accelerator, and here at the end the electrons are accelerated. I mean, in this linear accelerator, the electrons are accelerated close to the speed of light, and at the end it has a bunch of magnets that shake the electrons, and when the electrons move, produce coherent light at short wavelength. So this is a free electron laser. So these are facilities that need hundreds of technicians to work and millions of dollars in investment to build. And people go to these facilities with the experiments to do experiments. They write proposals, they go to the facility, and they run the experiment in a given time. So the rationale for our center was to invert this flux and fabricate portable sources that can go to the experiment. And this is the idea. In our center, we demonstrate short wavelength compact lasers. These are the lasers that we demonstrated in CSU. This is what we call the tabletop system. This is a capillary discharge laser. It's basically high voltage discharge that produces a very hot plasma, and the plasma emits a very strong amplified beam at 47 nanometers. A smaller model is this that we name desktop laser. The laser here is that big. It's the smallest UV source that you can find anywhere. With these two lasers, we demonstrate all the applications that we'll show today. And this laser works with a very fast, I will be very brief with here, but I want to give you an idea how this laser works. It's a very fast discharge in a capillary, in a ceramic tube. The ceramic tube is represented here. This yellow thing is a plasma. So when the current flows in the plasma, that produces a magnetic field. The magnetic field, by Lorentz force, compresses the plasma in the center, and they pinch the plasma approximately from 4 millimeters to 80 microns in about 40 nanoseconds. That produces a shock wave, and this shock wave ionizes the argon 8 times. That produces what we call neon-like argon, and this ion has this very strong emission line at 47 nanometers, and this is the laser. That's simple. These are interferograms obtained head-on, and this boundary here is the plasma sheet compressing towards the center, and in the collapse we obtain a very powerful short laser pulse. We utilize this pulse for many applications, and the nice thing is this laser is highly coherent. And this is a very simple experiment. It's a young double slit experiment, and you see that the interference fringe has a very large visibility, indicating that the laser is highly coherent. So it's very nice to utilize in applications where you need coherence. For example, holography, one of the applications I will describe today. So why we are going to UV? Well, it's very well known that in an optical system, the resolution is something that depends on the wavelength, the numerical aperture of your optics, and a constant that depends on many factors, right? So reducing the wavelength is a direct path to increase the resolution. And this is the motivation to use these lasers for the applications that we describe. One of them is microscopy. We replace the illumination in a microscope by our laser, and this is an example of an image of a half-wave fabrication microprocessor from AMD. These are lines, 100 nanometers, separated by 100 nanometers, and they are very clear, and they were obtained with putting the chip in reflection mode. This is just reflecting the laser in the chip and obtaining the image with a zone plate. The zone plate is our microscope. It's one lens microscope, one zone plate. Another application is lensless imaging, and this is something that we made in collaboration with a group of Margaret Murnane and Henry Katkin. And what we did is just shoot the laser through an aperture. And the idea is, when you have an aperture, you shine a very coherent beam that produces diffraction, and taking the diffraction of this image, you can reconstruct the object. Sorry, you can reconstruct the object. And here's the example. This is the object, this is a reconstruction, and this is the image. So let's go to the first application. The first application I want to describe is a very old optical principle that is used, that we use for a technological application, which is a nanopatterning. And the application is, the principle is the Talbot coherent imaging. So I will describe two approaches for nanopatterning. One is holographic printing, the other one is Talbot coherent imaging. In the first one, the holographic printing, what we did was to fabricate a hologram. We computer designed a binary hologram. We fabricate with electron beam writing the hologram, and we shone the laser through the hologram. This is an online reconstruction, and this is the reconstruction in the photoresist. These are atomic force microscope images of the print in the photoresist. With this approach, it's possible to print arbitrary patterns. These are test patterns, but you can do whatever you want, in a non-contact, mask-free system, very simple, with no optics, that in this wavelength at 47 nanometers is very important. This is very, yes?
I was just curious, when you do these exposures, do you have to integrate over multiple pulses?
Yes, we expose this for 200 pulses.
What is it like your typical rep rates or pulse lengths?
The pulse length of the laser for this laser is 1 nanosecond. Okay. The rep rate can go up to 12 hertz. Okay. So it's very short exposure. Okay. These things, for example, this was a proof of concept. I mean, we can go with much better resolution. This feature has 4.8 microns, and the resolution in this edge, in this case, was about 220 nanometers. Well, this is holographic lithography. It's called projection lithography or holographic lithography. More interesting is the Talbot coherent imaging. And the Talbot imaging is using a very old principle discovered by Talbot in 1836. we are specialists to taking all ideas and applying to new technologies. And Talbot, in those days, he had a grating that Fraunhofer gave him. And he was studying the grating with a microscope, and he put the image plane in the grating, and he saw beautiful lines of the grating fabricated by Fraunhofer. And because he has nothing to do, and there were no video games or TV whatsoever, so he was in the lab, no? And he started moving the microscope back, and after a certain distance, bang, the grating appeared again. And because he didn't have the Nintendo whatsoever, he kept going, moving the microscope back, and again, another image, and another image, and so on. These are called the Talbot self-imaging, or Talbot planes. And he also noticed that this grating appear very nicely, very precisely at given planes, and change the color when he moved the microscope. So he, of course, recognized that that depends on the wavelength, which is true. And this is a very common effect that you observe when you illuminate periodic structure like a grating with coherent light. What we did was to, instead to use the grating from Fraunhofer, we fabricate a two-dimensional grating. And the two-dimensional grating was an array of tiles, and the tile was arbitrary. In this case, were this test pattern, and we replicate these tiles in 2D in a large area, like 600 by 600 microns. So we had like a two-dimensional diffraction grating, right? And we shone the laser through this grating, and bang, at the Talbot planes, we obtain, these are atomic force microscope printings in the photoresist of the image, of the Talbot images that we produce with a mask. These are large areas. This is an example of a zoom, and these are another two examples. In this case, again, this feature from here to here is 4.8 microns, and here the resolution was about 110 nanometers. The setup is extremely simple. That's the Talbot mask. Here we remove the sample, the photoresist, but you put here your sample, you shine the laser through the photoresist and through the mask, and that's it. We replicate this, and we observe this replication Talbot images at up to the 6th Talbot plane. For this mask, the Talbot distance was 1 millimeter. So we were able to print areas like 600 by 600 microns at a distance of 6 millimeters with a resolution of 100 nanometers, which is pretty impressive. Also, I like to mention, these are, for example, the mask, and these are the printing in the photoresist. I like to say that this is equivalent to, I don't know if you are familiar with a technique for nanofabrication called nano-imprint. The nano-imprint basically is a very rudimentary idea, is to fabricate like a mold in a hard material. You fabricate your nanostructure in this mold, and you put a plastic or a soft material, and you press this mold against the material, and you transfer the nanostructure into the surface, right? And this is called nano-imprint. The guy who developed this technique was in 1998, and I think in the last 12 years he published like 600 papers in the area, so it's a big boom, right? So I like to say that this is a optical, non-contact, nano-imprint with one more detail, which is defect-free. And the reason is, in the nano-imprint, in the real nano-imprint, if you have a defect in your master, you will faithfully replicate the defect every time you print. However, if in our mask, if one of these legs is missing, because the area of the defect is negligible relative to the area of the mask, this is defect-free. So this is something promising, and I like to yes.
It has to be clear.
Yes.
As you go further ultraviolet to higher...
You lose resolution because the numerical aperture decreases.
So why do we use the 6th order?
We try up to the 6th. No. We use these are first order. We try at the 6th order to see how good we could print, and the 6th one, the 6th order, is well. This is look, Rafael. First, second, and third order. You see that it's getting blurred, right?
Yeah, the central ones are going to be good.
What?
Those in the center are going to look better.
Yes, that's also right. Yes, those in the center has larger numerical aperture, effective numerical aperture. Yes. This is the image at the 6th Talbot plane. I mean, it's blur compared with the first order. However, it's useful, still useful.
I have a question about
Yes.
how sensitive is your xeon then in that first Talbot plane?
In the second, here.
Okay.
Your question was very tiny. So you have two things. One is the resolution, and what you say is the depth of focus. And the resolution depends on the periodicity of the mask. This is P. W is the size of the mask, and lambda is the wavelength. For the depth of focus, this is the depth of focus is basically lambda over the numerical aperture square, right? Like in any system. And it depends on P square and omega. If you punch these numbers for example, a period of 1 micron, which is very reasonable to make, using wavelength at 13 nanometers, which is the new laser that we are developing in CSU, for example, right? And with a size of 1 millimeter, which is not easy to fabricate 1 millimeter membrane, but it's possible. The resolution in the 5th Talbot plane is about 8 nanometers. The drawback in this technique is that this is a 1-to-1 projection, right? So if you want 8 nanometers in your printing, you have to make if you want 8 nanometers in your printing, you have to make 8 nanometers in your mask. And this is not trivial, right? Yes?
Do you start with the exact mask you want and take the Talbot images, or do you ever use the sub-Talbot images where they're contrast reversed?
No. We look in the middle. You have the phase conjugate Talbot. Yes, we observed that. I didn't show the images here. But all these prints were obtained in the Talbot planes. So I just want to show this. It's not exactly imaging, but however, it's an imaging technique that we apply to nanofabrication. So, well, this is summary for this part. With the tabletop lasers, because they are so coherent, we were able to accomplish this application that needs very coherent sources. And also, they need high flux, right? These printers were obtained in 1 minute exposure. You can do something similar, for example, in the synchrotron, but you have to filter so much that probably to print a photoresist will need an exposure of 12 hours or so. So let's go to the second application. This is holography. And in holography, I want to mention two experiments. One is an experiment where we were able to deconvolve the 3D information that is in all holograms. And the second is to push the resolution down to the wavelength resolution. The scheme we use to take these holograms is the Gabor configuration. So the laser is in line with the object. The part of the light is diffracted in the object that produces the object beam, and the rest going outside the object is the reference beam. The superposition produces the hologram. The hologram is printed in a photoresist. The photoresist is read with an atomic force microscope, and this is, for example, one hologram from an atomic force microscope tip. So this hologram were reconstructed using a numerical code. This is the hologram, this is the reconstruction, and this is a zoom of in the reconstruction. And here you see the pixels. The resolution of this image was about 400 nanometers. to increase the resolution, what we did was to increase the numerical aperture. So we went with our object closer to the hologram, and this is the hologram. This is the reconstruction. After reconstructing, we noticed that the hologram, that the tip of our object was contaminated and broken, the tip. And the resolution in this image was measured doing a cut here and measuring the 10 to 90% change in the intensity was approximately 160 nanometers. The other experiment, and let's go in detail to the first experiment, is to try to obtain from the hologram the 3D information that every hologram carries, right? And to demonstrate that, we fabricated a special object, and the special object was a transparent membrane. This is a 100 nanometer membrane of aluminum that was placed between two uneven surfaces. So the membrane curved to these two plateaus, right? So basically we obtained a tilt transparent surface. Underneath we put the hologram. And on top of this membrane we put nanospheres, latex nanospheres, which are completely opaque to this wavelength. So my object now is a bunch of spheres, opaques, placed at different heights from the hologram. The hologram is here, the spheres are located at different distances. So this is what represents this image. This is the hologram, and what we did was to reconstruct the hologram using a Fourier transform code, backpropagating the image. So we took this hologram, we reconstruct the hologram, and this is one reconstruction. And in the reconstruction you can see that, for example, this guy here, if we make a cut in this direction, this guy is blurred. If we make a cut, this is what we obtain. It's very broad. However, this guy here is sharp. And if we make a cut, this is what we obtain. Why is that? Well, because in the reconstruction code, the distance that we plug in the computer for this object was correct. This one was out of focus, right? So what we did was to scan different distances and locating the different spheres, right? For example, this is an example for these two spheres. This one has a peak at 160 something microns. This one has a peak at 170 microns. So locating this number gives us the z coordinate, the xy we obtained from the hologram, and that's it. With the 3 coordinates we were able to plot or to reconstruct the surface of the tilt membrane. For example, we made the reconstruction in 2 different regions, close to the edge where we expect to have a larger slope, and out farther from the edge where we expect to have a lower slope, and indeed we obtain this, we reconstruct these surfaces. The other experiment was how good can we be our holograms. And to do that, there's no secret. The way to increase the resolution is to increase the numerical aperture. You know that. So what we did was to put the object close. We put a membrane close to the hologram. The hologram is a photoresist, okay? We put the membrane and we shoot the laser through the membrane. The object produced this object beam, represented here by these circles, and the flat is the reference beam. The interference is the hologram. In our case, the object was a carbon nanotube with a thickness of 30 to 50 nanometers. So we took the hologram, and this is an example of one carbon nanotube. The carbon, this is the hologram, and this is the reconstruction. If we make a resolution test of this object, the resolution indicates that this slope is approximately 45 to 38 nanometers, so close to wavelength. We did a more precise analysis of the image, and the more precise analysis is the following. It's a very complex method, but I will try to summarize quickly how we did to assess the resolution of the image. This is our image. So what we did in the image was to take a threshold and remove this low frequency noise. After that, we do the skeleton of the image with a code that takes the maximum of the pixels in this image. And with this skeleton, we fabricate the template. And the template was fabricated doing the convolution of this skeleton with a dot of a given diameter. And this is one example, this template. This template has a resolution by definition of 1 pixel, because our zeros and 1 is a binary template. So next, we took the template and we blur the template using a Gaussian filter. So we basically spread the intensity of the pixels in the neighbor pixels, and we produce templates with calibrated resolution of 1 pixel, this is the first one, 2, 3, 4, and so on pixels. Once we have this set of templates, we did the correlation between each one of these templates with the original image. And obtaining the maximum correlation will tell you that this particular template is the one that is more alike to the original image, right? And this is our measure of resolution. We can assess the resolution of the image. Doing this, we obtain plots like this. These are the different templates, and these are the correlation numbers. And for this case, the maximum peaks at 47 nanometers. So the image that coincides with a very simple cut, right? So it are two coincident results. So the resolution of this image was wavelength resolution. These are more examples of holograms of the nanotubes. The nice thing of this technique is possible to obtain images of large area, like millimeter square. In each sector you look at the image, the resolution is 50 nanometers. One more, and this is the last. I want, yes?
How did that compare with the resolution you get according to the numerical aperture? Basically the size of the hologram.
Well, with the theoretical resolution,
Based on your experiment.
yes, okay. Well, the maximum, absolute maximum resolution that you could obtain for this experiment was 32 nanometers. That's the limit with the numerical aperture of the exposure and so on. We degraded the resolution because in the middle we had to read the hologram with the atomic force microscope, so we have the convolution with the tip of the AFM, and the resolution of our image was degraded to 46 nanometers from 32. That was your question, Rafael?
Yeah, no, I'm still wondering why you used that deconvolution and blurring to...
Oh, because why? Oh, because
if you could just take two spots and try to resolve it,
we did give the same number. We developed this method because we like it, and it was fun to develop. we thought it was interesting, and they give it.
Kind of deconvolution.
What?
It's a type of deconvolution.
Yeah. Yes.
Did your hologram consist of kind of etching into the PMMA? Is that what it was?
Exactly. Yes. When the PMMA basically is a photoresist which is activated by photons. When you shine photons in the PMMA, the region that receive photons become more soluble to a solvent. So basically, you have the surface of a PMMA, and you shine the light in the surface. Okay. The region where receive more photons after developing are erased, are removed. So basically, what you obtain is in the photoresist is a level profile which coincides with the intensity profile.
Okay. So it was developed after.
Yes.
Okay.
Yes. 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 So let me show you the last result, and this is fairly new, and is our experiment using another kind of laser at shorter wavelength. This laser emits in the region of 13 nanometers, and this is a little more complex system. It's not a tabletop, but a several tabletops laser, right? It's like 4 tabletops. It's like a lab-size laser, right? So this laser emits at wavelengths close to 13 nanometers, which is also a good achievement, because if you consider that the scaling to go down in wavelength, scale with lambda to the 5th power, to reduce the wavelength in a factor 2 means that you have to increase the pumping power by a factor 2 to the 5th, which is not trivial, right? The only way to achieve this shorter wavelength is to shine a very high intense short pulse visible laser in a target, produce a hot plasma, and this plasma emits the short 13 nanometer laser. So with this laser we implement a microscope to inspect the mask that will be used in lithography at 13 nanometers. In fact, we demonstrate 2 different microscopes. One with a desktop system at 47 nanometers, and these are examples of the images. These are the one I showed before, the microprocessor. These are gratings that test grating to assess the resolution of the image. With the 13.2 nanometer laser, these are images that we obtain. These are also test patterns, and you see that the resolution here was about 38 nanometers. So let's go a little more in detail. What is the motivation of this work? And the motivation is everybody knows the Moore's law. Moore's law says that every 24 months, the number of transistors in a chip must duplicate. And this is the prediction of Moore in 1965, and the industry pretty much followed that, right? What is the limit here is what is called the critical dimension. The critical dimension depends on a constant that is called the process constant, depends on many things. One is the coherence of light and so on, the wavelength and the numerical aperture. And people so far had used different tricks to increase the numerical aperture and reduce the wavelength from visible to violet to ultraviolet, and now the industry is using 193 nanometers to print the chips. However, these things are getting more and more complex. The critical dimension limit around 32 nanometers is what is the limit, absolute limit, that the technology that is applied now in the manufacturing as a chip. So to go further in the shorter or smaller critical dimension, the semiconductor industry decided to make a big jump from 193 nanometers to 13 nanometers, and the next generation of chips will be printed at 13 nanometers. That's why this laser at 13.2 nanometers is so technologically important. And this is the way they are going to print. They will shine light at 13 nanometers into a reflective mask. The mask here is what has the circuit. So this is a mirror that will produce an image of the circuit, and after 6 projection mirrors, you will print with the magnification 4 to 5 times into your sample here. This is nice. I mean, there are like 6 machines like this that can print at 13 nanometers in the world, only 6. Each one costs about $90 to $100 million. It's very sad to say that 4 of them are in Asia, only 2 in this country, but there are only 6. Next generation will be with these machines. The thing is how to do the quality control of this mask. This is basically the heart of this process. And this is the mask. It's a 6 by 6 inches mask. These are the circuits. And the mask is basically a mirror. It's a multi-layer mirror, right? And on top of the mirror, you write your circuit. Now, this mirror should be very high quality because any defect in the mirror will print in the circuit, right? And you don't want that. You can inspect the mirror using visible light, no problem. And you will detect only defects on the surface. Why? Because visible light will penetrate zero in these multi-layers. The multi-layers are silicon molybdenum, so it's completely opaque to visible, right? So with the visible light, you can see these defects. But there can be defects like this that are called buried defects in the multi-layer. And the only way to detect these defects is to go with wavelength that penetrates the whole thing. And the only wave that penetrates the whole multi-layer is 13 nanometers. So you have to inspect the mask at wavelength. And this is the secret here. The options to inspect this mask is to use visible and UV scanning. It's fast, it's very common, it's technologically developed. However, you cannot see defects, phase defects, or buried defects. You can use directly the printing tool. You can go with the mask into the printing tool and see what happened, right? But this is very costly. If the mask is bad, you have to go back, right? It's better to inspect the mask before going to production. You can do actinic scanning. So what you do is just to very quickly raster scan the mask to find defects. And you can detect this with actinic, it means at 13 nanometers. You can do that, but this is a completely different scheme to locate the defect, and the other is to determine if this defect will be printable or not. And to do that, you have to do a full field actinic inspection. And here is where our laser has the capability to make very precise high-resolution images of actinic at wavelength of these defects. Imagine how complex is this problem that to detect a defect that should be is around 15 to 20 nanometers. These are the size of the printable defect. In a 6 by 6 inch mask, is equivalent, scaling the problem, to find a baseball in the state of Colorado looking through the state, right, to scan the state from an airplane, right? This is the difficulty of the problem, right? So it's not simple. So going from actinic inspection, there are two machines. For example, now one is in Berkeley. It's basically the beam from the synchrotron is shown in this machine. You put the mask here, and you move the beam in the sample and in the mask, and try to locate the defect. The same thing with the new Subaru ring. This is an accumulation ring that produce also coherent light, and they obtain these two systems are not working. We use our laser, and this is a scheme of the laser. It's a little more complicated than the one I showed before with the discharge. It comprises 3 amplification stages in a titanium sapphire laser that is shined in a target. The target is illuminated with 2 powerful beams that produce a plasma, and that plasma emits the coherent light at 13 nanometers. I will not go into details here, it's not useful. But we fabricate with this laser this microscope. And the microscope has 2 lenses. One is the condenser here, and the other is the objective here. And we mimic the geometry to make exactly the same incidence angle that the mask will utilize in production. It's 6 degrees here. So this, let me show how this microscope is built. This is the condenser soap plane. Basically, this is the soap plate, and this is an aperture. The soap plate is to focus the laser beam into the sample, and the aperture is to let the image go through, right? So this is a window just to let the image go through in the reflected mode. The objective zone plate is this. The zone plate is in this region. Actually, is a section of the zone plate. The zone plate is that big, but only this part is printed with the lines of the zone plate. The numerical aperture is the effective numerical aperture. The resolution is limited by the numerical aperture of this region. With this laser, this is the laser, and this is the chamber with the microscope. You have the mask, the objective, and the condenser here, and the laser beam comes in this direction. It's very compact. The microscope is something like this. It's a vacuum box that size, right? This is the object that we tried with different test patterns. And we had two different kinds of images. One is a dark field, which is a reflected lines in the absorber sample, or bright field, which is absorber lines in a mirror. So using this mask, these are the images that we obtain with the microscope. These are test patterns with different periods, and these are cuts of this test pattern, and you see that the fringes are very well defined, very well resolved. If we measure the contrast of these images for all these periods, we were able to fabricate or to plot the modulation transfer function of the microscope. These are the data points, and extrapolating the resolution of this microscope was about 40 nanometers, a little less, 38 nanometers. So the other thing that we studied was how homogeneous was the illumination. And this is a critical thing because laser is coherent, and you know that when you image with coherent light, you have fringes, and this time you don't want, right? So we were able to shake and move the condenser, superposing many pulses and blurring the interference pattern, and having a very nice and homogeneous illumination, and the line roughness of the printing was shown here. Is very nice. These are images of two gratings with a very homogeneous illumination. So finally, I want to speed up. It's too long. Finally, the estimated resolution of the microscope was 55 nanometers. However, this is conservative. We think that our resolution is below 40. This microscope allows exposure with very short exposure, and the exposure depends on the mask, and depends on the particular experiment that we are running, but always is very short exposure, and we were able to find or to obtain a very uniform illumination, having very nice and homogeneous images of this lithographic mask. So I want to acknowledge all my collaborators from CU, Berkeley, and Colorado State, and overall to these guys who are the responsible of many of the results all the results that you saw today. These are the students in our group. Do you have any question? Yeah. Yes.
Is the source for this last laser you're discussing, is that the tin plasma?
No, it's not the tin plasma. The sources with tin plasma are incoherent.
Okay.
These are the sources that are developing now the industry for fabrication. I mean, these big machines are made with a liquid tin plasma. So
CO2 laser.
Exactly. They have droplets of tin, and they intercept with a CO2 laser, and they produce a bright plasma. But this emits 4 pi, right?
Okay.
So you have to collect all this, and you have a lot of debris with these sources, and this is a big problem now to solve. How to mitigate the debris of the plasma that accumulates in the collecting mirrors,
Okay.
right? And how to improve the emission of this wavelength for make the source reasonable to print. These are, the 13.2 nanometers is cadmium. It's a solid target of cadmium, and we shine 2 pulses for reasons I will not explain now, but it's like 15 minutes of explanation.
Okay.
2 laser pulses, and this produces a elongated plasma column, and this, under certain conditions, is a laser medium, and that amplify photons along the axis at 13.2 nanometers.
What kind of coherence length can you get?
The spatial or temporal?
Temporal.
Temporal, about lambda over delta lambda is 10 to the minus 4.
Okay.
And spatial is very lousy, I would say 50 microns or
Okay.
so.
All right.
Yes,
I'm assuming you're using zone plates because there's no traditional optics for these.
that's right.
Do you have any issues in terms of like, you know, you're throwing away a level light there? Does the laser's high enough power to do that? I mean, I guess what I'm asking is like, do you have any issues in terms of the amount of light getting through the zone plates and going to like the first order focus?
Well, that's a very nice question. Yes, the efficiency of our zone plates is about 10%,
Okay.
right? Which you say, well, you are losing 90% of the light. Yes, but fortunately we have a lot of light,
Okay.
so still is good.
What sort of power outputs are you getting out of it?
Depends on the laser. In the last laser, the 13 nanometer laser, the power, the energy per pulse is about 16 microjoules.
Okay.
For the other one, the 47 nanometers, it can be up to 1 millijoule.
Oh, wow.
Yes, wow. Yeah. In fact, when we made the experiment with the lensless imaging, right? The setup is very simple. It's just the aperture, and we shine the laser through the aperture. And the first time we put the laser through the aperture, we blow the aperture out. We evaporate the aperture. So, yeah.
Do you have any issues with like interference of light from the second and third order focuses?
No, because the second and third order, the efficiency is 1% and 0.01%. Yeah, no.
What? I'm sorry, I was just kind of curious what kind of system transmission efficiency you got for the last system, the 13 nanometer.
For the 13 nanometers, the throughput, I mean the attenuation of the. Let me see.
Just the system transmission, like
Well,
the efficiency was.
The 45, the first folding mirror is the reflectivity is about 70%, and and we have one condenser and one objective, and for the condenser we have a 10% efficiency, and for the objective we have 10% efficiency in the zone plate, and we lose, because the membrane where the zone plate is placed, we lose like 40% of the light. So I never made a calculation, but you have the numbers.
Okay.
Yes.
Remind me, is it 14 nanometers the water window?
The water window is 8 to 4.
8 to 4.
Yes.
So you're not there yet.
Oh, we will.
So 13 is a lot better than it was, but you're still not transparent.
13 is very nice because this particular application in lithography, right? And this is technologically important because next generation will be print at 13.5. And we have 13.2, which is...
Oh, I'm thinking ahead to biological applications.
Well, we too. And
Yeah.
I give you an update where are we. For the 13, the 13, this is the mirror reflectivity, and 13.5 is the peak, and the industry select this wavelength because this is the best mirror that you can fabricate in this region. That was the only reason. But we are good. Here we can use our laser, right? Going to biological application would be nice to have water window, absolutely, yes. And we are now developing a tabletop system that will work, hopefully, at 8 nanometers. And hopefully means that probably 1 year down the road, after hard working, we will be able to demonstrate a system like this. And we also plan to go with all the expertise in microscopy that we accumulate with this 13 nanometers into 8 nanometers to biological application. Yes, of course, yes, that's the... No?
Questions?
Okay, thank you.